Today it's my pleasure to introduce to you Professor Alan Bergland. So Alan has been an assistant professor at the University of Virginia since 2016. And prior to that, he did his postdoc at Stanford University with Dmitry Petrov, his PhD at Brown University with Mark Tatar, and his undergraduate research with Bill Bradshaw at the University of Oregon. So his research has focused on the genetics of phenotypic plasticity and local adaptation in Drosophila and Daphnia. And he has a particular emphasis on the role of seasonally varying selection pressure on the maintenance of functional genetic variation. And with that, I'll leave the floor to Alan and I thank him to have agreed to present at the same.
Thank you so much. Now let me get screen sharing going here. All right. So thank you very much for that kind introduction. And today I am happy to talk with you about the research my lab has pursued studying the genomic basis of rapid evolution in fluctuating environments. So my research is motivated by the basic question, why are individuals different from each other? This question is central, of course, to evolutionary biology and also central to the study of all living systems. We address this question by working to characterize the strength of evolutionary forces that shape patterns of genetically based phenotypic variation at the phenotypic level and also at the molecular level. For instance, we ask whether genetically based phenotypic variation is shaped by stabilizing selection, right? In which deleterious variation is removed from a population by selection, or rather if diversity is affected strongly by diversifying forces, in which variation is maintained by selection. At the molecular level, we characterize the scope of genetic variation within and among populations, and in particular, we're interested in identifying conditionally beneficial mutations that are good in one environment, but not another. So the standard neutral model of molecular evolution, as well as the classical models of evolution, emphasize the importance of stabilizing forces acting on populations. And as a consequence, most genetic variation segregating in populations is either neutral or mildly deleterious. And under this classical model, conditionally beneficial mutations are very rare. On the other hand, if diversifying forces are common and layered on top of that neutral model, we might expect much more variation in the genome to be maintained by some form of diversifying selection, thus arguing that, that diversifying forces are an important evolutionary force shaping genetic variation in populations. All organisms live in environments that vary across time and space. And in my lab, we study how temporal and spatial fluctuations in selection pressure maintain genetic variation underlying fitness-related traits. In my lab, we address this basic question using two organisms, Drosophila and Daphnia. Both of these species or species assemblages are broadly distributed across the world. And that allows us to study adaptation across spatial gradients and also to study the repeated evolutionary process that occurs on different continents. Both of these species also have a very rapid development time, and that enables them to have multiple generations per year, allowing us to study the evolutionary dynamics in real time in response to seasonal fluctuations in selection pressure caused by a number of different factors. In my lab, we study these questions by trying to understand the genetic architecture of traits, the genotype-phenotype relationship, by working to understand the evolutionary dynamics of alleles and also populations using genomic inference. This work seeks to identify environmental drivers of rapid and cyclic evolution. And we address these basic topics using tools that we develop, computational tools, as well as some physical tools that we develop. And our work benefits and also contributes to community and citizen science-based efforts. So since I've started at the University of Virginia, my lab has pursued several major projects. And I want to just orient you to the first two very quickly. These are work that we pursued in Daphnia. And this is work of Dr. Karen Barnard-Cabot, as well as Dr. Dorothy Becker. And I'm not going to have time today to go into the full details of these projects. You can read more about them. There's preprints available. But I do want to just tell you very briefly what the punchline of these stories are. So this first project, seeking to examine polygenic variation in sexual investment in Daphnia, focused on a set of populations that we studied in southern England in the Dorset region, a meta-population consisting of a series of ponds that vary in the degree of ephemerality. Some of these ponds are deep and permanent, and some are very ephemeral. Some are intermittently ephemeral. And using whole genome sequencing of about 500 clones that we sampled in this population, we were able to reconstruct a pedigree and show that this pedigree is consistent with the degree of ephemerality. In permanent ponds, you have a single clone which tends to dominate for long periods of time. In very ephemeral ponds, you have a much more genetically diverse set of clones. And then in this intermittently ephemeral pond, you have periodic rise to higher intermediate frequency of a limited number of clones, followed by complicated mating relationships. And we were able to show that these two dominant clones that existed actually differ in their reproductive investment strategy. And we've done a lot of work to try to identify and characterize attributes of the genetic architecture of this variation and show that variation in male production rates seems to arise rather rapidly in these populations and is subject to strong fluctuating selection. The second project that we worked on with Daphnia sought to understand the evolutionary forces affecting variation in phenotypic plasticity. So Daphnia, of course, is famous for growing these pedestals and spikes on its dorsal edge in response to predator cues. And using clones that we had sampled, about 100 clones we had sampled from these ponds, some of which were genetically unique, we exposed these clones to chiromone, to predator cue, and using a high throughput imaging analysis that we had developed, we were able to characterize the shape along that entire dorsal edge. And by contrasting patterns of heritability as well as mutational variation between the clusters, the genetic clusters, as well as between the environments, we make an argument that standing genetic variation in plasticity is lowest in areas of greatest plasticity, suggesting that there is strong stabilizing selection operating on this trait. All right, and I'm happy to talk with folks more about that if they would like to. All right, now the remainder of my talk, I'd like to tell you about our work studying Drosophila and studying this phenomenon of seasonal adaptive tracking. So to motivate the study of seasonal adaptive tracking in Drosophila, I want to talk about different ways we can think about seasonal adaptation. So on the one hand, we can view seasonal adaptation as phenotypic plasticity, right? This is changes in behavior and physiology that allow organisms to escape the unfavorable season, right? Either escape it in time or in space. And these sorts of phenotypic, these plastic responses typically evolve in organisms that have long lives or are able to migrate long distances. On the other hand, you can view seasonal adaptation as a process of adaptive tracking. And that can occur in organisms that have very rapid generation times and maybe limited dispersal capacity. And under adaptive tracking, the idea is that there potentially is a winter adapted genotype and a summer adapted genotype that have alternate fitnesses in these different environments. And as a consequence, the allele frequencies underlying these different genotypes fluctuate through time. We study this process of seasonal adaptive tracking in Drosophila. Drosophila is a great model to study this process. It has a large population size. Regionally, it's probably on the order of 10 to the 5th or 10 to the 8th, depending on what scale of geography you're looking at. They're multivoltine, so they have multiple generations per year, and they're highly fecund, enabling populations to grow very rapidly over the course of the summer. They're also globally distributed and of course are easy to collect across different habitat types. One of the habitat types that Drosophila lives in are orchards, and orchards, of course, go through seasonal changes in resource abundance, as well as temperature and other environmental stressors. The basic model of Drosophila metapopulation biology and seasonal biology is that populations start at a relatively low density early in the growing season. And then over those 5 to 10 generations that occur, the population grows until winter comes and then it dies back. And over that period of winter and early spring, populations potentially eke out a couple of generations, although we don't know exactly. If you go out and sample flies at different times of year and bring them back into the lab and rear them in a common environment, you can assess genetically based differences in phenotype between flies collected at different times of year. Work primarily from Paul Schmidt's lab has shown that there is a number of different phenotypes that change over the course of seasons. So for example, flies collected in the early part of the growing season in a mid-Atlantic orchard show higher starvation tolerance than flies collected in the fall. And the idea here is that those flies you collected in spring are the descendants of those individuals that survived winter, whereas the flies you collect in the fall are the descendants of those individuals that did really well in the summer. So patterns of starvation tolerance, the propensity to enter diapause, as well as aspects of pigmentation, amongst others, show these patterns of seasonal variation. These patterns of seasonal variation are consistent with a basic allocation model, right, in which those flies that survive winter invest more heavily into somatic maintenance, also a trait that we think would be common in the north, whereas flies that do really well in the summer that you would collect, right, towards the end of the growing season would be those genotypes that allocate more into reproductive output. If you were to identify alleles that underlie this basic life history trade-off, a winter favored allele in blue and a summer favored allele in red, and measure those allele frequencies through time, right, you would see potentially that they would be changing in frequency, and that over multiple years, there would be an oscillatory pattern of allele frequency change. So to test this basic idea, we sampled flies in a Pennsylvania orchard over the period of 3 years during the spring and fall. And using pooled resequencing, we identified alleles which show this pattern of oscillations in allele frequency. So each of these lines in this plot is a single SNP, and what we're showing here are fluctuations across time. And we argue that this was a basic signature of seasonal adaptive tracking occurring in this population. We wanted to ask another question, which is whether seasonal adaptation is a general feature of fly populations living in temperate environments. And so to address this question, as a postdoc, I helped organize a meeting at the NSF-funded Nesson Center. And the purpose of that meeting was to develop a collaboration of Drosophila biologists across North America and Europe to sample flies near their home institutions over multiple years. So this is the data set that we produced. This data set is called the DROS-RTEC data set, Drosophila Real-Time Evolution Consortium. And this map shows sampling localities in North America and Europe, where we have paired spring-fall samples for either 1 or 2 years. And I'm going to talk about some inferences that we were able to make using what we call the core 20, which is 20 pairs of spring-fall samples that are collected across a number of different localities. In some cases, we've included data for up to 2 years. Using this data set, we fit for every SNP in the genome, GLM, a linear model, where we regressed allele frequency on season. And then we permuted the data, permuted the spring-fall labels, and reran that GLM across every SNP in the genome. And what we're showing here is it's a representation of a p-value distribution, showing the number of SNPs in a particular p-value bin. And what I hope you can appreciate is that in the real ordering of the data, there's many more significant SNPs that are showing association with spring-fall seasonal labels than we would expect under permutation. And using this piece of evidence, we can argue that seasonal adaptation is a general feature of fly populations across different temperate locations. Next, we wanted to ask whether we can predict allele frequency change in any one population. To do this, we performed a leave one out analysis, and this uses the core 20 set. And in this core 20 set, what we did is we sequentially, we broke it up into a discovery set composed of 19 populations, and then a test set of the remaining population. And in the discovery set, we identified seasonal SNPs using that same GLM. And for any given seasonal SNP identified in the discovery set, we can ask whether the direction of allele frequency change was the same in the test set. And we can then do that sequentially dropping each population. What we then do is we can ask for SNPs that are significantly seasonal in the discovery set and the test set below some significance threshold. What fraction of SNPs show concordant allele frequency change? What I'm showing you here is the result of one population. And as you can see, as you increase your significance threshold, so looking at the top 1% of SNPs in both the discovery and the test set, you see that about 57% of this allele frequency change is in the correct direction, in a consistent direction between the discovery and test set. And for this particular population, that concordance rate increases generally with increasing significance thresholds, suggesting that there are consistent allele frequency changes between this particular test set population and the discovery set. When you look at all populations, you see that many of them show this consistent sign of allele frequency change, meaning that each of these individual populations looks like the average of the rest. But there's a curious thing in this plot, right? There are 4 populations which showed a reversal, right? And this reversal, the way to interpret it is that for these 4 populations, allele frequencies in the spring look like fall and vice versa. And we wanted to try to understand what was driving this, this reverse concordance or this strong discordant signal. And the way that we approach this is that we assign each population a prediction score. And that prediction score is colored here, the color of these lines, and it reflects the slope of this, this line, how strongly you become concordant or discordant with increasing stringency. And we sought to try to identify environmental features that could predict the heterogeneity in prediction score. And to just get to the punchline, what we found is that localities that had warm springs and cold falls showed reversals. So using publicly available weather data, we calculated the number of days in spring when the temperature was above a given threshold, and the number of days in fall when the temperature was below a given threshold. And we had built this model under the hypothesis that a reversal should be caused by a particularly warm spring or a particularly cold fall, making the reversal possible. And what we found was that if you can calculate the number of days in the 2 weeks prior to fly sampling where temperatures were above about 30 degrees Celsius, and number of fall days where temperatures were below about 5 degrees Celsius, you can predict variation in this, or you can explain variation in these prediction scores. You can explain about 75% of the variation in these prediction scores. And these values of T max, of temperature max and temperature min, correspond roughly to the thermal limits of flies that we know of. So this model is consistent with something that we know about fly physiology. So can we predict the allele frequency change in these populations? Well, the answer is yes. Recent temperatures can predict the sign of allele frequency change from spring to fall. Next, what we wanted to ask is whether we can predict phenotypic evolution within and among populations using genomic data. And to address this question, we wanted to work on diapause as a model trait for seasonal adaptive tracking. So just to tell you a little bit about fly overwintering, Drosophila melanogaster, like other Drosophilids, overwinter as an adult. And diapause in these organisms can be assessed by looking at patterns of reproduction. And commonly it's assessed in females by looking at the number of mature eggs and the telegenic oocytes that they are harboring in their ovaries. And under normal breeding conditions during warm days, flies will make many eggs, right? And when you expose flies to cool temperatures, if they undergo and enter diapause, they will not make any telegenic oocytes at all. I previously showed you this slide, which showed that the genetic propensity to enter diapause changes through time and also between habitat types, as well as the genetic propensity to enter diapause changing across latitudinal gradients. This work that I'm going to talk about is the work of a postdoc in the lab, Priscilla Erickson, and she wanted to ask several basic questions. Is the genetic architecture of diapause Mendelian or polygenic? And also, how does the landscape of genetic variation of diapause change across time and space? And one of the things we wanted to do initially was to confirm that genetic variation in diapause was subject to temporally varying selection pressures. And so to do this, we built an experimental orchard where we planted peach trees and then encased them in these big fly cages. And we built compost piles and released genetically defined populations of flies in these cages and tracked a diapause propensity over time. And this was at a field site near UVA called Morven. What we did is we collected flies from these cages, brought them back into the lab, and reared them in a common lab environment under different temperatures, and contrasted the propensity to enter diapause with control cages of flies that were maintained indoors. So what you're looking at here on the y-axis is the proportion of flies in diapause, and these are all in G2 flies. So these are assayed in a common garden using granddaughters of females collected from cages. What you can see is that the outdoor cages, flies show an increased propensity to enter diapause when temperatures start to get very cold, suggesting that diapause is actually associated with survival of cold in the field. Whereas we see no change in the propensity to enter diapause of the control cages that were maintained indoors. So this was good evidence, of course, that genetic variation in diapause is subject to temporally varying selection pressures. What we wanted to do next is to understand something about the genetic architecture of diapause. Now, oftentimes in Drosophila, when you are studying the genetic architecture of traits, you might be tempted to use the Drosophila genetic reference panel, which can be a useful resource. But what we wanted to do is we wanted to understand the genetic architecture of this trait in an outbred population with the idea that it might more accurately reflect levels of standing genetic variation seen in the field. What we did is we pursued this using an approach that we call a hybrid swarm, where we use inbred lines collected up and down the East Coast. We did a whole bunch of round robin crosses to make F1s, and then put bunches of F1s in these large cages where they were allowed to intermate for multiple generations. We then took this outbred population of flies and distributed them across environmental chambers that we built that varied in temperature and photoperiod. So one advantage of this approach was that we were able to assay fly populations across about 48 different environmental combinations of temperature and photoperiod, which is something that would be really hard to do in a standard inbred mapping panel. We then took individual flies after they had been exposed to these different environmental treatments for different periods of time, and we assayed them for diapause. And then we performed very shallow sequencing followed by genome reconstruction to perform GWAS. This is an image of these cages, and as you can see, they are just crawling with flies. These cages probably had a census size on the order of 50,000, and we had to wear protective clothing to go in and get our samples. This mapping process is made possible because of our ability to very cheaply and efficiently reconstruct whole genomes. Because we know the genomes of these inbred lines that went into the founding cages, with only about a 0.5x coverage, we can reconstruct fully phased and very accurate genomes, roughly about 99% accurate, using very sparse sequencing data. So that's one of the things that we developed that made this project possible. So the first thing that we found was that diapause in our mapping population has a moderate but non-zero heritability. So using genetic relationship matrix type approaches, we can show that the heritability of diapause is on the order of about 12%, and much greater than we'd expect by chance. These gray points here represent permutations. Next, what we show is that diapause is polygenic. There seem to be many loci in the genome that are significantly associated with diapause above what we would expect by chance. We wanted to take what we know about phenotypic distributions of diapause across time and space, coupled with the natural history data that we have, right? The allele frequency estimates we have of populations sampled through time and space, and test the basic hypothesis that the pro-diapause alleles should increase in frequency across latitudinal climes and also should be at higher frequency in the spring compared to fall. And we tested this hypothesis by calculating polygenic scores, which are just the sum of the coefficients from GWAS and clinal or spatial models. When we look across multiple populations, what we see is that when we contrast our populations sampled up and down the North American East Coast, that we see evidence supporting what we had hypothesized, that populations in the North are more likely to show, populations in the North are more likely to harbor the pro-diapause alleles compared to populations in the South, and that beats permutations. However, our core 20 multi-populational seasonal model does not show any noticeable difference from the permutations, and if anything shows sort of a curious reverse relationship where you potentially see slightly more or slightly fewer pro-diapause alleles in the spring compared to the fall. This was particularly interesting, this seasonal result that we had. And the weak signal, but also the potential reverse signal. And so what we did is we broke our populations into each of the individual paired spring-fall samples. We calculated that polygenic score. And what we observe is that for some populations, they show a strong positive value consistent with what we would expect. And for other populations, they show a strong negative value. So what it appears to be is that the direction of allele frequency change from spring to fall in these populations is somewhat idiosyncratic. So can we predict phenotypic evolution within and among populations using genomic data? Well, yes, to a degree, but predictability varies due to local conditions. All right. So the next question we wanted to ask is the fundamental question that many of us are after, which is what are the genomic targets of seasonal adaptive tracking? And to address this question really requires a lot more data than we had been working with so far. And so I organized along with Drosophila, which is a very well organized consortium of people in Europe and also a little bit throughout the world now, who have been studying Drosophila evolution and sampling populations collected in their home institutions for long periods of time. And together we put together the Drosophila evolution and space and time data set. And this data set integrates three main Drosophila data sets: the Drosophila Genome Nexus, which is a compilation of genomes of 900 individuals produced by a number of different groups. previously published and unpublished Drosophila samples. So these are pool-seq samples collected by the Drosophila, as well as the Dros-Artex samples that I told you about previously. Collectively, this data set represents 13,000 flies, sequenced in total to over 16,000 X coverage, representing 271 unique pool-seq population samples. And it's one of the largest temporally resolved population genomic resources available to date. And it's really been a fun project to work on and a fun consortium project to work on. This project includes a pipeline for reproducible and scalable bioinformatics. So a pipeline that's going to allow us to grow this data set as more populations become sequenced by our groups as well and our consortium, as well as other groups in the field. We wanted to make this data set available and accessible. And so there's a website that you can go to where you can download not just the final files, SNP files in the format of VCF files and other formats, but also intermediate files that you might be interested in. There is also good source code and well-organized metadata so that you have geographic and temporal information for each of these samples. And again, this program was a global collaboration, which was really fun to work with, and a number of members of my team participated in the development of this project. Now, I just want to highlight a few of the interesting findings from this work so far. One is just a basic characterization of Drosophila population genetic structure. These are principal component projections of allele frequency data. And what you can see is that the Drosophila populations roughly cluster into 4 groups: the North American group over here, African populations, and then 2 European clusters. And these European clusters had also been previously identified in the Drosophila paper that came out in 2020. So these 2 European clusters form an Eastern cluster and a Western cluster that are very easy to resolve on principal component 3. And one of the things that we did in this project was that we used model-based inference using moments, which is a 2D site frequency spectrum-based approach, to estimate the split time between these Eastern and Western European populations. And we can estimate that split time to be on the order of about 1,000 years. So this work not only provides this interesting resource for the Drosophila community, it also provides insight into Drosophila population genetics, and also provides some resources for doing demographic inference using pool-seq data. And we've already extended this data set out quite a bit. We have been collecting flies really diligently in Charlottesville, where we're at the University of Virginia, at an orchard. So this is a apple and peach orchard, which you can go and get fruit at, and the university is right over here, not too far away. Alyssa Black, a PhD student in my lab, collected flies every 2 weeks over the period of 3 years at this orchard. And we wanted to do this really dense collection in order to perform a more fine-grained genotype environment association. And using weather data from a weather station at the orchard, we characterized weather in the days prior to sampling, and then calculated the average temperature, as a characterization of the environment. And we regressed allele frequencies at every site against the average temperature to identify SNPs that are correlated with temperature. Following this SNP level GWAS approach, we performed a sliding window analysis where we broke the genome up into 100 KB windows and asked how many SNPs in that window are in the top 5% of these p-values for the GLM. And we use this to calculate then a window-wise p-value. And this identifies linked clusters of temperature-associated SNPs. What I'm showing you here is a slide which focuses in on chromosome 2L. The y-axis in panel A is this window-wise p-value showing the difference from our null expectation of 5%. These windows are colored by the proportion of SNPs in them that are in the top 5%. And what I hope that you can appreciate is that there are these large clusters of SNPs that are strongly associated for temperature-dependent allele frequency change. These SNPs are inside a large cosmopolitan inversion called IN2LT, and they're also enriched near the breakpoints as well as in the center of this inversion. Using some other window-based metrics, this is a Stouffer's Z method of combining p-values. You see a similar enrichment. And we can show that these SNPs that are inside the inversion are in reasonably strong linkage with each other, right? But are not in perfect linkage with the inversion breakpoints, but are in pretty high linkage with the inversion breakpoints, and suggestive that perhaps these loci inside the inversion are what are driving allele frequency change in that whole region. And we've been able to use the wealth of Drosophila GWAS data out there from the DGRP to then try to identify whether these regions that are associated with temperature-dependent allele frequency change are also associated with phenotypes, right, measured in the DGRP. So what I'm showing you here are the number of phenotypes associated with number of phenotypes where you have an enrichment of SNPs that are associated with temperature and also associated with traits. And generally speaking, these regions overlap, right? The phenotypes that are associated in these regions tend to be behavioral phenotypes and activity-based phenotypes, suggesting that temperature-dependent behavior may underlie some of the seasonal fluctuations in allele frequency that we see. So we're interested in pursuing this question, whether IN2LT is a supergene. And we're excited to address these questions in Drosophila. We think that we'll be able to test hypotheses about modes of action. For instance, we'll be able to ask whether it's the inversion per se that's under selection, or whether the actual targets of selection are some form of co-adapted gene complex residing within the inversion. We'll be able to test hypotheses about phenotype, right? So I told you about some hypotheses that we had generated using DGRP data, but the other benefit of the system is that there's been 30 years of research on IN2LT and its effects on temperature-dependent aspects of fitness. And then, of course, we'll be able to use Drosophila genetics to identify causal alleles associated with phenotypic variation. All right. So what are the genomic targets of seasonal adaptive tracking? Well, at this point in time, we think that it is potentially linked to an inversion polymorphism, and I look forward to telling you more about this story as we unravel it in the months to come. All right, so I want to just wrap up by telling you why I think this work is important. First, I think this work is important because it shows that fluctuating selection is an important evolutionary force that is a diversifying force in populations that can drive the high abundance or the abundance of conditionally beneficial mutations. Second, I think that this work in Drosophila is important because it's a really tangible example of evolution in action. This is a picture from my fruit bowl at home, Drosophila sitting on it. And when I tell people about the work that we do, oftentimes the question I get from them is, how do I get rid of fruit flies in my kitchen? So people are intimately familiar with fruit flies and their seasonal dynamics. And we've been working to actually capitalize on this public interest by developing a citizen science project where we're working with residents to collect fruit flies from their compost piles throughout the course of the summer. And we're using these collections across the state of Virginia, coupled with a broader sampling at different orchards, to try to understand something about the seasonal dynamics of fly populations, the seasonal dynamics of the Drosophila community, and to understand the evolutionary differentiation between microhabitat types, between compost piles and orchards, and to get this really fine-grained metapopulation view of seasonal dynamics. And so we've been working with an organization here in Virginia called the Master Gardeners, and we work with them to, we send them these fly collection kits and informational material, and then walk them through this process. And we're now in the 3rd year of this project, and have actually received quite a large number of flies from them. Finally, I want to argue that that this work is important because adaptive tracking is probably a general feature across much of life, right? So if you think about animal biomass on Earth, the vast majority of animal biomass is composed of small things with relatively rapid generation times. And thus, under this view that short generation times, multiple generations per year, is one of the key features of seasonal adaptive tracking, it's likely that many organisms out there are engaging in this process, as well as other forms of diversifying selection. All right, with that, I would like to thank the members of my lab at the University of Virginia, as well as collaborators in Europe and North America, as well as our funding. And with that, I will be happy to take any questions.
All right, thank you. So does anyone have a question that they can write on the Q&R tab? Or perhaps, meanwhile, our host, Romain, has some question for Alan?
Yeah, do you hear me?
Yes.
Yeah, thank you for this very interesting talk, Alan. I really enjoyed it. Yeah, actually, I mean, you kind of answered a bit my question, but I actually, when I was looking at the first part of your talk and when you present your 2014 paper, and then you see that a lot of SNPs are actually moving in the same direction. So the if you have like a polygenic character, you I mean, with selection goes less fast than with inversion. And so, so my initial question was, oh, could this be inversion that are locking multiple traits, or that explain all those you know, this SNP, all those convergence of SNPs across different time points, which you in part answer in the last part of your talk. But so yes, my question is, do you think this this temporality is usually widely associated with the structural variation?
Yeah.
Or...
Yeah, it's a great question. So in that initial paper, the signal was very distributed throughout the genome, and we had made an argument that it was strongly polygenic, and we saw no enrichment with inversions. In the Drosophila paper, which had more data, these 20 paired string-fall samples, we showed that the signal is distributed throughout the genome, but it seems to be enriched near inversion breakpoints, including 2LT. And then this last study that I told you about, where we focused much more specifically on temperature, we see a strong signal in the inversion, but basically very little signal elsewhere. I didn't show you the genome-wide plot, but there are some little blips that, up here and there that we think are possibly associated with pesticide resistance. Yeah, that's the basic answer. But remember that the last study that I talked about, we focused on a single environmental variable. And so we might be missing a lot of SNPs that are associated with other aspects of the environment. So it's probably not exclusively 2LT, but 2LT is probably a major driver.
Great. And do you know, I mean, it's I'm not very familiar with Drosophila inversions, but you mentioned it's a cosmopolitan inversion, so it's associated with possibly some other traits like pesticides or temperature or.
Yeah, that's been the mystery for 50 years. It's been associated with temperature in the past, and you can actually, there's been work looking at experimental evolution in cages at different temperatures, and 2LT fluctuates. So that's what we do know some about it, but we're working to try to piece together what's going on there. It's yeah. So I can tell you more in a few months. How about that?
Yeah, that sounds cool.
Mathieu Joron?
Hi. My question was if you had populations that were fixed for one or the other inversion forms, and or are you in the middle of a decline or something?
So most populations are polymorphic for 2LT. Most so they are at it's actually the most sort of common inversion, at least in North America, and it shows a very, very weak cline, which is probably actually consistent with it being affected by temperature. And so you wouldn't necessarily expect when you went out to sample that it would be at the perfect sort of linear relationship along the cline. There is a signature of clinality there, but it's not as strong as some of the other inversions. But it's polymorphic pretty much everywhere.
So do you think this is the reason why this inversion is having such a shallow cline?
Yes, that's exactly right.
Okay. Thank you.
A kind of a small comment or question myself, what I found fun with this story is that some of the earliest examples that I know about fluctuating selection tract live in nature was actually about inversions, I think by Dobzhansky about some 80 years ago, also about season. So do we know, and it was in another species, I think it was Drosophila obscura, I don't know, can't remember. So do we know if, do we know more about this? It's kind of a curiosity question about what's in those inversions that linking, I don't know, specific traits relating to seasonal adaptation. Do we know more about this?
Yeah, it's a great question. So I don't know if anyone has, I mean, there's been a lot of work on the inversions in Pseudo-Obscura over the last 10 years. I don't know if anyone has gone back to really investigate the seasonal dynamics in that system. People have been working pretty extensively on Sub-Obscura in Europe, where there's a really interesting like allelic series of different inversion types that have been shown to actually respond to heat waves, that change in frequency in response to heat waves. So there has been work examining seasonal fluctuations in inversions. You're right, starting back from Dobzhansky, but still to this day, but in other Drosophila species.
Okay. And do you plan to have in some of these populations to have a more a denser temporal tracking than what you did, or is it feasible?
Yeah, so there are some populations in the data set that is part of the DEST data set already, which have more dense sampling. We are continuing to go out and sample very regularly. And like I said at the very end, we're sort of extending our dense temporal sampling to other orchards in the region, as well as these compost piles. So we're hoping to build out a more dense sampling network. And then I think that folks, especially in the DROS-EU, are oftentimes able to sample more densely in some cases than others. So there are more samples out there, and certainly it is key. So in some of the DEST data set where we have more samples, we do see a hint of stuff going on in 2LT, but it really does require, I think, very, very dense sampling to see it.
Yes, my question was also implicitly what I meant was, do you have, it would be interesting to see if there's some lag between the waves or the waves of frequency and the seasonality, as you were saying. The traits that you see have been selected, the traits that you see often have been selected in summer, etc. So there has to be some kind of lag. would be interesting to investigate that. No?
Yeah, absolutely. Absolutely.
Other questions? Someone? Friday afternoon for holidays for some people? People from the organizing committee? Nick Planidine, okay. Raise your hand, so let's see. All right, Nick.
Hello.
Hey.
Hi.
Okay.
Yeah, we can hear you.
Awesome. My question is about the predictability of the like spring fall allele frequency changes and how you see some populations have that inverse relationship. So do you think that like with a particularly mild summer or winter or extreme, that would be due to like a plastic effect where they can just buffer against the environment within a generation, or it's just reduced I guess intensity of selection that is flipping the trend there?
Yeah. So what we sort of put forward is that this is selection causing changes in allele frequency, and that, like you said, that either a particularly mild spring, which would cause allele frequencies to rise, or a particularly cold fall, which would push allele frequencies into a more winter-like state, which is what we think is the selective agent, or what we think is the process underlying that reversal. It's entirely possible, right, that what's happening is some sort of temperature-dependent plasticity, right, perhaps in behavior, and so we're actually sampling flies differently at different times of year, and maybe there aren't selective deaths. But that's kind of an open question that we're trying to explore. So it could be plasticity, it could be selection. So far, the data seem to be consistent with selection. But yeah, does that answer your question?
Yeah, no, that's great. Thank you.
Mm-hmm.
More questions? No? Okay, so then in that case, anyway, some people have booked some timings to talk with Alan at the beginning of next week. So I just want to thank Alan again for this great talk. And let the speakers, the audience know, sorry, that we're having a 2-week break for the school holidays, and then we're coming back on March 11 at the usual time, 11:30. Still don't know if this will be in person or only on Zoom, but we'll let you know in due time. So with that, have a nice weekend, and thanks again, Alan.
All right, thank you all so much.